Welcome to the second epigenomics exercise of CSB250. Today we're going to be extending on what we learned last time and making it genome-wide. So we'll start at the genome browser, which is something you're probably used to by now. And the thing that we're seeing right now is epigenetic marks across a given locus. And what we want to do now is say, how often do we see these epigenomic marks come together across the genome and also to integrate additional data. So to do that, we're not going to be working in the UCSC genome browser, but instead we're going to be using a tool called Galaxy. And Galaxy is available from Penn State University, and when you get to this website you'll see something like this. And what this is it's a series of tools built into a web browser, so you don't have to run this locally, it all runs online. And these series of tools allow you to grab epigenomic data, or really any genome-wide interval-based data, and to work with it. So the first thing that we want to do is, in this case, if we're going from where we had, in this case, K562s, which are a cancer cell line, and we want to see if I have marks of an enhancer activated right now, which is K4 monomethylation, K27 acetylation, and not K4 trimethylation. This is one example right here of something that is marked by those two, but not the third. And so what we want to do is be able to get all of the data on these tracks. And in this case, we don't want the actual signal data right here, which is these blue kind of histogram plots, but instead we want to deal with the peak data. And this is chromosome start, stop, and we want to be dealing with just those intervals, and that is in interval format. So we will go back, in this case, to Galaxy, and what we'll do is go over to Get Data. And Galaxy is integrated with the UCSC genome browser in order to allow you to just pull in data from, in this case, UCSC, from what they call the table browser. So you'll activate this screen right here, and we're working in the human genome, in this case HG19, which is the most recent build. And we want to go to find groups. Now groups in this case correspond to the different tabs that we've been looking at previously, and we want to go into regulation. In this case for ENCODE histone marks. And to do that we'll go to the group is regulation. The track is under Broad histone, and then the individual tracks, in this case they list them as tables. We want to select down and find K562s, and in this case we want to search for H3K4 monomethylation, and we want to look for this PK, or a peak file, not this signal file. We want to be dealing with just the peak, so just the interval format. You can leave the rest the same, and the only other thing you need to change now is to go to the output format and go to selected fields, because we don't want all the information that's coming with. We really just want to get chromosome start and stop, and that's all we're going to need for this particular data set. So we'll hit done, and you can send it to Galaxy. And what it's going to do then is it's going to send it over into this pane which kind of holds all our data, and it's going to have a little clock while it's waiting. It's going to turn yellow while it's working, and you'll have this little spinner, and once it's finished it's going to turn green. There you go. So this is going to give you information about this particular data set because you imported it from UCSC genome browser, but for our purposes we're going to want to keep track of it in a little bit more detailed way, or a little bit less detailed way. So you can go to this little pencil and change the name, in this case to be K562 H3K4 monomethylation peaks. What you have down here is a little bit more information about this data, and you'll want to make sure, because we want this to be an interval format, that it's recognizing that you have chromosome start and end. If you click on this data in this pane, it will open this up and it'll tell you how many intervals or regions that you have, what genome build it's from, and it'll give you a little preview of your data. So right now you have chromosome start and end. And Galaxy has recognized that these are the three different column types that you have. If these aren't recognizing chromosome start and stop, it'll throw an error. So all you would need to do is go in and just change these to let it know which column that's actually in. Okay, so we'll save this. And now we have K562 H3K4 monomethylation. For the sake of time, I'm not going to go through and get the H3K4 trimethylation and H3K27 acetylation, but what I want to do as part of integrating data is to look at whether or not the DNA methylation of these potential enhancers has any sort of enrichment. And so to do that, the other thing that I would like to get for this is to go over back to the UCSC genome browser, and instead of histone marks, I want to go to DNA methylation, and this is from the Hudson Alpha Institute's DNA methylation profiling with chips, and we're just going to go to K562 and take selected fields. In this case, this is a chip that profiles individual CPG dinucleotides and uses a bead-based assay to see if it is, or what percentage or signal strength is methylated or not methylated. So we'll go to chromosome start and end, and without getting too much into the internals of how this assay works, you also want to know the score, and you can read more about this on the UCSC genome browser if you click through, because it'll explain the data. But a score of less than 200 approximates to something that is unmethylated, and something greater than 200 is methylated. So we're going to use Galaxy in order to filter through that data so that we can keep it in separate categories. So here we're done, and we'll send this over here. So while this is working, I'm going to sign off, and we'll pick it up on another video to start to integrate and overlap this data with each other.